I'd like to welcome everyone to Sernova CEO conference call. Thank you very much for joining today. My name is Dr. Philip Talakis, President and CEO of Sernova Corp, and with me is Dominic Gray, Head of Communications at Sernova. We are pleased to host the CEO conference call today following Sernova's annual general meeting of shareholders held earlier this morning. A recording of this conference call will be made available after the call on Sernova's website. I will first provide a quick introduction to the company for investors and then provide a corporate update as we're moving forward. The presentation will be followed by a question and answer period, and to facilitate the fluidity of the call, participants have been muted. So let's start with the presentation. Excellent. These are forward-looking statements. First, what I'd like to do is talk a little bit about the regenerative medicine field. And as you probably know, Sernova has been in the regenerative medicine field for over 10 years now. So we are quite advanced relative to quite a number of other companies. So if we examine the regenerative medicine field, there are a number of different approaches. One is to provide organ regeneration. The next one is gene therapy approaches. And the third one is cell therapy approaches for therapeutics. And Sernova is mostly focused on cell therapy with some work that we're doing in the gene therapy area. So why is regenerative medicine important? We find that it's a real paradigm shift in the treatment of chronic diseases. Regenerative medicine itself provides the potential to have what we call a functional cure as opposed to masking disease and long-term treatment of symptoms with prescription medicines. So Sernova is very well positioned in the regenerative medicine field for success. We have intentionally focused on a stepwise approach, first learning about our cell pouch with human donor islets, and then moving forward with our stem cell derived technologies. And this dedicated approach had led to success and leadership in the field. We have seen a number of regenerative medicine companies assuming that they can get over the cell therapy technical barriers with a home run type approach. And we've seen a number of failures in the field. And we're really proud to say that Sernova has advanced in the field. And we believe we are producing excellent data from that perspective to prove what we have done. So with respect to Sernova being an innovator and leader in the field, we are a publicly traded company, both in the US and Canada and Europe. And we are a clinical stage company with a clinical trial ongoing at the University of Chicago. So we have a cell therapeutics approach platform. Our platform uses an integrated regenerative medicine approach, as we've discussed. We're working with our implantable cell pouch, and we have immune protected therapeutic cells. We have a broad platform that we're working with. We are working in multiple large market indications. Everyone is very familiar with our diabetes indications, and you can see from our recent press releases that we are advancing our hemophilia field as well as our thyroid applications. And the cell pouch itself has been very carefully designed to overcome the barriers with therapeutic cell survival and function. And our cell pouch forms an organ-like environment after it's placed deep under the skin in the body. So before the cells are actually placed into the cell pouch, a beautiful organ-like, highly vascularized environment, typical to what you have in organs, is ready to accept the cells. And those cells will be placed into the chambers of the cell pouch. And once they will engraft into the tissue, they will produce their missing proteins or hormones to be able to treat disease. Diabetes is our lead program. And we have the active phase 1/2 clinical trial ongoing. And we've also conducted proof of concept studies that were successful in both hemophilia A and thyroid disease that we'll talk about later. I'd like to go back and talk a little bit about our Sernova's platform approach. So as we know, the cell pouch has been developed to be able to form this organ-like environment after it's placed in the body within a couple of weeks. And that provides an ideal environment for the placement of the therapeutic cells. And our therapeutic cells can be human donor cells, such as the one we are currently using in the clinical study, where we're using the real thing, which is human donor islets for the treatment of type 1 diabetes. And we also have a stem cell derived program where we can take stem cells and then drive those through a process into islet-like structures, and we can work with stem cells also for the hemophilia field. And then these cells and tissues produce and release the missing hormones that are required to treat disease in the bloodstream. And we've proven that we can do this with a number of applications. The third part of the stool, essentially, is immune protection. So how do we protect those cells from attack by the patient's immune system? So as everyone knows, our patients for our clinical trial have been taking the immune protection medications at this point in time. And that's allowed us to advance our clinical study to be able to show that we can get our insulin into the bloodstream from our cell-pouch islets. So this has been very important. But as you know from today's press release with Ajax, that we are actively working on local immune protection technologies. And we're going to talk a little bit about that later. So we're really proud to be able to be moving these processes forward from that regard. With respect to our patent portfolio, we have international patents and patent applications around the world. And this includes composition of and use of medical devices for delivery and cell transplantation, glucose responsive insulin secreting stem cell technologies, and local immune protection technologies. So really importantly, Sernova has patents in North America, South America, Europe, and Asia. We don't talk about these a lot, but this is a lot. There's been a lot of work in the background prosecuting patents, and we're really, really proud of our patent portfolio. And why is this important? Because as we're working with pharmaceutical companies and we start the process of potential licensing and this kind of thing, then these companies require us to have a strong patent portfolio. So if we talk a little bit about diabetes, we're now 100 years past the time when insulin was discovered. And if you think about that 100-year period, patients have been taking insulin by injections, either a needle or an insulin pump. As far as we're concerned, very little advance has been made in the field. And if you look at the reality of diabetes, there's over 400 million people affected worldwide, and 10% of these have type 1 diabetes. And this represents a commercial opportunity of over $30 billion conservatively for Sernova. And our hope moving forward is to find a functional cure for everyone suffering from diabetes. And what we have seen over, with a number of different companies is that there's a lack of a real integrated regenerative medicine therapeutics approach. And the future for Sernova is blockbuster potential for our platform to establish a new standard of care going forward. So Sernova's pipeline, I'm just going to be very brief on this. Our clinical study is in phase 1/2, so that's safety, and we're also looking at efficacy measures. Our program with our stem cell technology is working on a local immune protected program, and that's part of what we're doing with Ajax and some of our other technologies that we're looking at. The Hemophilia A program has worked with corrected patient cells. So we've actually worked with human cells that have been corrected for the production of factor VIII, and we've been successful in our preclinical models showing that we can get factor VIII into the bloodstream and have a positive effect on hemophilia A. And in addition, we have been working with benign thyroid disease, and I believe we're the only company in the world that is developing that has a thyroid program, where we have inserted human thyroid tissue in preclinical models and been successful there. So if you look at the market opportunities for Sernova, look on the left-hand side, and we have patents in Europe, the United States, North America essentially, and also in the Asian countries. So we have a global IP patent portfolio. And then if you look at the potential patient populations for 2020, and you're looking at severe hypoglycemia unawareness, which is what we're doing with our current patients at the University of Chicago, we look at there being about 4,000 transplants that are occurring. If we start to move with our stem cell derived technology, and we have the ability to treat approximately 1.5 million patients. And then if you look at all type 1 diabetic patients that we're able to treat, we're looking more at a population of approximately 10 million patients. So then if you look at the right side, in terms of the potential opportunity, our initial opportunity is around $400 million for a year once we get the product cleared. And then if we move into our stem cell derived technologies with the hypoglycemic unaware population, then that's about a $24 to $30 billion market. And when you combine that with all type 1 diabetic patients, then you start to see a market that is significantly higher than that. So obviously there's enormous potential, and this is only for our diabetes program that we're working on. So if you have a look at the competitive landscape, we're laying out the major companies that are out there, and that includes Sernova. You've heard a lot about ViaCyte. You've seen the success of Sema with respect to that preclinical company being purchased for $1 billion Canadian. And also Sigilon is in the background, but also doing work in that area. They're a company that's been started recently. If you look along the top, let's just talk about who has efficacy data, who has clinical efficacy data showing C-peptide levels in the blood. And as you know, C-peptide is a biomarker that is a gold standard biomarker for proving that insulin has been released by cells in the body itself, right? So our islets produce insulin. They also produce C-peptide at the same time. That C-peptide gets moves into the bloodstream. So if you look at device vascularization, islet engraftment, that has been demonstrated in humans. We'll talk a little about that. And then local immune protection technology is required. And then we'll talk about the financial side of things. So Sernova obviously is the only company in the world that has shown C-peptide levels in the bloodstream with our device and therapeutic cells. No other company has done that as far as we're concerned. With respect to device vascularization and islet engraftment, we have demonstrated this in all of our preclinical models over and over again, and we've now demonstrated in the clinic in 2 clinical studies that we have done. ViaCyte has failed originally in their first program with respect to vascularization, and now they have been working on trying to develop a device that has pores in it that allows blood vessels to get in and giving their patients immunosuppressive drugs, right? So they're moving forward. And we don't know very much about what Sema has done or Sigilon from that perspective. We haven't seen data there. For the local immune protection technology, Sernova is aggressively working on bringing in these technologies right now, and we're looking at owning the technologies. And the Ajax agreement that we have is a significant advancement in that with respect to that area. The local immune protection at ViaCyte, we're not sure how that's working, but their current program requires long-term immunosuppressant drugs. If we go to the financial markets now, you look at comparison between the different companies, and our market cap is approximately $29 million US as of today. ViaCyte has raised money to the tune of $240 million US. Sema has been sold for $950 million US as a preclinical company in its field, and Sigilon has raised almost $200 million. So you can do the comparison metrics with respect to the successes that have been proven out in the clinic and in the field. So really interesting, when we went to JP Morgan this past January, which was a different world, we met with a number of our pharma partners. And one of them, from one of the biggest companies in the world, quoted it, and they said that we thought the cells would be the hard part, and we focused our efforts on developing cells for diabetes. And he said, it is obvious now that having a functional device is the limiting factor for all companies out there. And there are very, very few options. And he named Sernova as literally the only company that has been proven in a clinic with our device. So what are the challenging issues, and what have we gotten past with our technologies? So first of all, scalability. We know that Cell Pouch is scalable. It was designed early on by our scientific team to be scalable. big issues with medical devices. When you put them in the body wants to surround them with a fibrotic sac. And we knew very early on that what we needed to do is create an organ-like environment. And we were able to not have this issue of massive fibrosis going on in our device with our device design. Vascularization has been another thing that companies have failed at, and we get significant vascularization because we have pores in our device that were developed early on. We've proven that we get cell engraftment, so that means that when you put the cells into the device, they're going into a tissue environment, the blood vessels connect up to them, and they're in a natural environment, so we've been successful there. And obviously our device has been proven to be biocompatible because it's made of materials that are already approved by FDA for permanent use in the body. And finally, from the versatility perspective, we've proven that we can put human cells in and be successful. We've proven that we can put tissues in the device and they survive. We've worked with stem cell derived technologies, and we've shown very good long-term efficacy in our device. So if you look at the therapeutic cells from that perspective, so we have been working with human donor cells, and that has enabled us to get early initial efficacy testing in the clinic. And you see from the results that we're starting to produce at the University of Chicago trial that we're getting really positive efficacy indicators that we put out in our press releases. And that's really allowing us to advance our technologies. The issue with these human donor cells is the supply is limited, but they enable cell pouch validation to occur as we're working on our stem cell derived technologies. We move to our stem cell derived technologies that enables an unlimited supply of cells to be produced for the large markets that we were talking about. And we have the ability as we're going forward to treat all patients with type 1 diabetes and also other indications. So what about these stem cell derived technologies? Does Sernova have any access like SEMA does and these other companies? Well, as you remember, a number of years ago, we put out a press release where we got worldwide exclusive rights to a diabetes stem cell technology from the University of Toronto, UHN to be specific. And this was licensed from patents and technologies were developed by Gordon Keller and Christina Nostro, who are luminaries in the field. So we are very, very proud to have our own stem cell derived technology, and that competes with all of the other companies in the world. We just haven't been making a lot of noise about this, but we're developing this in the background. With respect to other aspects, we also are looking at pharmaceutical collaborations, and a number of companies, as we have mentioned, have stem cell derived technologies. And we, because of our successes, are able to advance partnering opportunities. And the way this typically works is that in the regenerative medicine field, it's really important to be able to combine technologies and understand how these work and make sure that everyone is really confident prior to licensing. And so we have been working from this perspective too and going down this path very, very successfully. So if you look at the immune protection, there are a number of opportunities, and Sernova is really dedicated this year and right now to bringing in local immune protection technologies, because this is really going to expand our valuation and our possibilities. So we started out with our patients with immune protection medications, and this is working well. These medications have been around for many, many years, and it's known how to work with the patients so that they are safe and they're being monitored, and that's what we have with our clinical study right now. And all of our patients are doing well. Now, with respect to immune protected macro devices, I've talked about this many times. So if you take a medical device like the cell pouch and you try to protect that from the immune system, then what you have to do is close off all the pores that stops the blood vessels from getting into the device. And this immediately leads to the possibility of having massive fibrosis occurring in the device. So we stayed away from that approach. We separate our local immune protection, and we do that inside of our device. And this is one of the unique features that allowed us to be successful. So another idea is taking, for immune protection, from a local perspective is to take the cells and wrap them in a very, very thin polymer coating that has pores in it that allows blood sugar to get in, allows insulin to get out. And this is one of the technologies we see as very, very positive and something that could move to the clinic very quickly, relatively quickly. The next one is gene cell tolerance. So here, what you're doing is taking and editing the gene of the cell itself, so the starting cell, so that you can make that cell blind to the immune system. And this is the technique that we're working with Ajax. So this is a very, very advanced technology that we're now working with at this point in time. So the idea here is that we are looking at multiple approaches for local immune protection. And we're working with state-of-the-art technologies, state-of-the-art companies. So why is Sernova so unique? We have what we call a biologically relevant delivery process. So you can see, again, we place our cell pouch deep under the skin. You can't see it under the skin, you can't feel it under the skin. And within a couple of weeks, it fills in with the pores with these vascularized tissue. And then we go in at the right time, and we've proven that our device can go in after a couple of weeks, and then it can be stable for months and months and months down the road. And then we can put the cell therapy in when it becomes available. So then what happens is, as I said before, the cells will engraft. So the blood vessels will come up close to the cells. They'll feed them. And it will then allow the therapeutics product to be released into the bloodstream. Okay. It's not letting me. Okay, from a strategic development approach, I'll be very quick about this. So again, we work on our cell pouch device. We're able to use human donor cells, stem cell-derived cells, as well as tissues. And we have a diverse immune protection approach we're using. We're working with anti-rejection medications. We're looking at approaches such as being able to coat cells in polymers that can be able to protect those cells, and a gene editing type approach. But again, our systematic approach of working with human donor cells to start with and the anti-rejection medications has allowed us to get this efficacy. So with respect to our clinical progress, you can see that there are many, many, many activities that have to go on prior to, as you're moving forward from the clinical development stage. And Sernova's clinical team has done an absolutely outstanding job in getting Sernova into the clinic, working with our investigators and working with our CROs to do this. And as a result of that, we've been advancing our University of Chicago study. And I know everyone is always saying faster, 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 but what we're doing is we're doing this right, as we've done this all along. And doing it right means that you get really good success and things are done properly. So with respect to our first clinical trial, we focused on hypoglycemia unawareness patients. So these are the patients that have the biggest unmet need in diabetes, and it affects around 15% of type 1 diabetic patients. And what is hypoglycemia unawareness? Well, this is a situation where patient is unaware when they take an insulin injection, their sugar levels can drop to a very dangerous low level, but they don't get the shakes and tremors like people without diabetes get. So they can actually go into a coma. And if you think about it, every insulin injection is like playing Russian roulette. So these patients really need a therapy, and insulin doesn't work, and they need a cell therapy. And that's why we decided to work in this particular area first. So if you look at our phase 1/2 study at the University of Chicago, the primary objective of the study is safety. As we're moving through here, and that's safety of the cells, safety of the cell pouch together in this group of patients with severe hypoglycemic episodes. The secondary objectives, which are really important, is these are efficacy objectives. We want to see how the device is working, how the cells are working to be able to treat diabetes itself. So one of the efficacy measures is survival of endocrine tissue in the cell pouch. So why is that important? So in the system, and this is a new field, you want to make sure that the cells are surviving, that they're becoming highly vascularized, and that they're able to produce insulin long term. So this is very important to be able to prove. The second one is the proportion of subjects with a reduction in severe hypoglycemic events. So that makes sense because we're trying to help these patients out. And then we're looking at the reduction of HbA1c. And HbA1c is a long-term indicator of how your body is doing with respect to control of blood sugar levels. And then we're looking at 20 additional endpoints. So what's really important here is that this is not going to be a binary outcome study whereby it's either successful or a failure. We are learning about how the cells in the cell pouch work, and we're able to really see what kinds of measures and things the cell pouch and its cells are being able to improve with these diabetic patients. So this is a very, very important study from that perspective. So what is the status of the study? We've already said that we are treating multiple patients right now. And we've shown some positive efficacy findings that we have already announced, and we're continuing to enroll and recruit patients moving forward. And I just do want to mention and remind people that we have gotten significant support from JDRF. So our clinical protocol has been reviewed, and JDRF is providing grant funding for this study. And obviously, we're working with Dr. Witowski, who is a luminary in the field at the University of Chicago. So very, very briefly, I'm just going to go into this quickly. The way the protocol works is that we implant our cell pouch, and then for a number of weeks, and then we start the immunoprotection medications on the patients, and we stabilize them. So that's very new in the field. A lot of times what people do is give their cells to the patients, and then they give the immune suppression at the same time. And we believe that this is a much better approach to what we're doing. So what we then do is at the 90-day time point, we also have small little pouches that have the islets in them, and we remove those and leave the therapeutic pouches in, and then we have a look at those under the microscope, essentially, to see how the islets are doing. And we have these independently assessed by a pathologist to tell our team how the islets look and whether they're vascularized, whether they're in the tissue matrix, and this kind of thing. So this gives a good idea of what's happening in the therapeutic cell pouches. And then at 180 days, what is really good is that we have the opportunity to put a second dose of islets in there. So we typically, when you're doing a drug study, you like to be able to do what we call a dose response curve to see what's happening when you increase the dose of the cell. So we're learning about that in this study, and that's really, really important. So the study goes on, and the patients are followed up after they get their second dose of islets, and then we obviously take a huge number of efficacy measures and safety measures as we're going through the study. So we have released some initial findings from the study, and our safety findings so far are showing from that initial release that occurred by the physician itself in a conference that we had no incidences of adverse events. Cell pouch is very well tolerated. We did not get any fibrotic or other reactions to the cell pouch implant. And the physician noted that the cell pouches, when he went in to put the islets, was very well incorporated with vascular tissue and ready to receive the implant. So why is all of this important? You know, people think safety is not so important, but it's really important because it forms the basis for us to be able to use this product as a therapeutic moving forward. So when you look at additional early findings that we have, these are 90-day findings after cells and cell pouch were placed into the body. And what we showed was a reduction in hemoglobin A1c. This is what we were talking about before. We showed a reduction, almost 50%, of the need for long-acting insulin and here. And we've also importantly showed a reduction in the severe hypoglycemic events by 75%. So this is really very, very exciting from our perspective. So we wanted to allow some data to get out. As the study is going forward, it's very, very important to be able to collect the data and have that data be independently assessed. And there is a huge amount of data that is being collected at the site and then going to CTI. I know that we keep getting requests from investors to put out data as all the data comes out, and we're allowing data to be released at the right time to preserve the statistical integrity of the study. So this has to be done right from that perspective. The other thing we've shown at 90 days is really important, and I believe that we are the first company with a device that's placed under the skin that have shown and proven an increase in blood levels of the C-peptide, proving that we're getting insulin released from the cell pouch, going into the bloodstream, into the vessels of the cell pouch, and then going into the bloodstream. And this proves that we have increased levels of insulin. So our patients also are getting continuous glucose monitors that we've gotten from Medtronic. So what does that mean? That means that we place the glucose monitor on the patient and it measures glucose levels 24 hours a day for 14 days. And this occurs prior to cell pouch and cell pouch transplant, and then it occurs at different intervals throughout the study. And if you look at the top graph, in green is where you want your blood sugar levels to be able to stay. And this is prior to cell pouch, and you can see that the blood sugar levels are bouncing all over the place. And then at the bottom, this is 90 days after the implanted transplant. And what we're showing is considerable more stability. And really important for the hypoglycemic events is that you don't want these going down below the bottom of the green level here. And you can see that we haven't got any of those with cell pouch. So the conclusion was that we have what Medtronic called a stable glycemic profile with less excursions above and below. And if you look at the right-hand side, you can see that the high levels went from 285 milligrams per deciliter down to 231 at the 90-day time point. The low glucose levels were a baseline of 50, and they were increased to 66. So that gives us a more stable glucose response with cell pouch and islets. The number of glucose excursions went from 15 to 3, and the number of low excursions went from 8 down to 1. So that would be considered a real reduction in the hypoglycemic events, which is one of the key parameters of the study. So as we're moving forward, we're collecting more data, and what our hope is to continue getting these kinds of positive data in our patients. So we mentioned going forward here about removing the Sentinel device. And other companies have removed their Sentinel devices and found fibrotic tissue and dead cells. And if you look at this, we provided this from one of our patients to our independent pathologist. And he came back and he said there are abundant, viable, organized islet cells within the device. Those islet cells were intimately associated with blood vessels, which they absolutely need to be able to be survived. And they were sitting within a natural collagen matrix. And the islets were strongly expressing insulin. So it couldn't be better. And if you look at the right-hand side, this is a picture that shows in red islets, and those islets in red are stained for insulin, and the green are blood vessels that you can see going right around and through the islets. So these would be considered what I would consider beautiful pictures of islets within our sentinel cell pouch. So, so when you think about the sentinel cell or the cell pouch, this is what we're expecting is also in the therapeutic islets. And when you think about that, so you have your islets, they're producing insulin very strongly. There's blood vessels all around them. The insulin's getting into the blood vessels. It's then going into the systemic circulation of the body. And we're seeing production. We're seeing C-peptide in the bloodstream, we're seeing insulin in the bloodstream, and we're seeing improvement in our hypoglycemic unaware events and other efficacy indicators going forward. So we're very, very fortunate to have released this early data to be able to show how things are going. So just so you know, most clinical trials, what they do in companies is they'll start their clinical trial and you won't hear anything for 2 years. So we consider this very fortunate that the physician has allowed some of these data to be released. So if we go a little bit back in time, we have already conducted a first-in-human clinical trial up in Canada, and these were with similar patients that have severe hypoglycemic events. And here, with this first-in-human study, we again were able to show beautiful islets with the vascularization around those cells and in red production of insulin. And again, we met all of our safety criteria. So if you go back and look at our preclinical studies, so this is really like looking at the roots of a tree. And Sernova has probably got the strongest roots developed for a regenerative medicine company in the world. We have conducted large animal studies with diabetes and been successful there. And we're the first to show efficacy in 2 different large animal models. And we've also shown a proof of concept study in mice, and this has been independently confirmed by other investigators. So cell pouch being tested by other investigators have also shown we'd be able to get insulin independence. So if you look at the cell pouch and you look at the environment it's created, we've been able, and this is basically, if you take the cell pouch and you make a slice of one chamber, you can see the clear areas where the cells go, and then the tissue around it is the tissue that brings in the blood vessels. And we've proven that this can occur in mice, in pigs, and in non-human primates. And we've also been able to put the device in different parts of the body, in the abdomen, the limb, interscapular. So what you really want to do is be able to show how versatile your technology is. And this is really important as we're looking at various indications, working with other big pharma companies. You know, they may want to put the device in different places. And as we're working with different pharma companies in different indications that you may not even have heard of yet, our device can be put in different places in the body. So a little bit of preclinical data. So we've shown in a large animal model where we've taken the pancreas of the pig and we've isolated its own islets, placed it into the device itself, and we got efficacy after we proved that the animal was diabetic. So here in purple, you can see, again, within the cell pouch, islet, what we call islet clusters, and every cluster was associated with a blood vessel, showing that we are absolutely getting blood vessels and survival of islets. We also are showing on the right-hand side that we're producing these beautiful islets with insulin staining. If you look at the same slides, you can see that C-peptide is being produced because we know that C-peptide and insulin are produced at the same time. So this is validation, validation, validation. And finally, we've proven that all of the other hormones that are required are being produced. So the cells in the islets are producing insulin, different cells producing glucagon, different cells producing somatostatin. So if you think about insulin pumps are designed to produce insulin, and the very most advanced pumps are starting to produce glucagon. Well, Sernova's cell technology is already producing everything that you need. So I'm going to stop there and go on to our Hemophilia A program, right? So what is our goal there? So everyone knows that we have been working fastidiously, our R&D team, with a group of European academic investigators to develop a product whereby we take a blood sample from the patient that has hemophilia. So we're working with the real thing again. We're able to do what we call ex vivo gene therapy and replace the gene in those cells that is missing, that stops the production of factor VIII. So we now have cells that produce factor VIII. And what we did was we were able to work with our team to scale those cells up, ship them to Canada, and our R&D team was able to work with an animal model of hemophilia and be able to place those cells in the cell pouch, be able to prove that that they were surviving in there, that they were becoming vascularized, and that they were producing factor VIII, and that they actually improved the clotting in that preclinical model. So this is a very, very significant advancement. And as everyone knows, we just put a press release out just recently to talk about that our team was invited to the Gene and Cell Therapy meeting in Boston in a virtual meeting and given a podium presentation, which is very prestigious to be able to have, and presented the successful data there. So we believe, again, we're first in the world with this application moving forward. So this just shows a little bit of some of the biology here and some of the technical information that I'll move forward on. And with respect to our thyroid program, again, we've been very quiet about that, but been moving forward in that very aggressively. So we have received a grant that we worked with a group at the University of British Columbia. And here, what we're doing is we're again working with human tissue, and we've gotten permission to work with tissue that has been explanted from patients that are having their thyroid glands removed for benign purposes. And what we've been able to do is take that tissue our R&D team has and place it into the cell pouch, and so again, survival of those cells and production of the precursors of thyroid hormones in the bloodstream. And I believe that we are the first company in the world to be able to work on this treatment. And we have looked at the markets, and there's over 150,000 patients in North America are having their thyroid glands removed annually. And this represents a 2.2 billion opportunity. So why is this important? Well, these patients have to take thyroid medications every single day for the rest of their lives. And the important thing here is that if you've ever known a person that is missing thyroid, that they have depression, they have issues with weight gain, and the drugs just aren't satisfactory. And we're looking at, again, as we are in diabetes and hemophilia, working to replace the function of the thyroid gland that has been removed. So look forward to more information coming out on this program. So again, this one just shows some of the tissue successes that we've had with that thyroid gland in the cell pouch. So beautiful proof of concept going forward. So we're kind of running out of time here. I could probably talk for another hour about successes of Sernova and where we're going. But what are our next steps? So we are, even with today's announcement, we're working on working with companies to examine local immune state-of-the-art protection technologies. And we're also looking at licensing these in so that we own the technologies and we're able to work with these to the clinic. So you'll see this happening. We are also expanding our worldwide, our multi-family patent portfolio. So the idea is that when you get a patent, you have 20 years when you file your patent application, and we have filed additional patents from that, in that regard. So we work very, very hard with our patent lawyers to expand our patent portfolio. Our goal for this year, and this is pending all of the things going on with COVID-19, we're hoping to complete our study patient enrollment, and that means implantation of pouches in patients. We're working very hard to be able to do that within the context of COVID in our clinical site, and then continue on with our patient treatment and follow-up and safety and efficacy evaluation. With the Hemophilia A program, as we have said, we presented results at a very prestigious conference and gotten kudos from the scientific world from that perspective. And we're looking at developing the market and product positioning, and also looking at the regulatory and clinical plan and developing a path moving ahead to the clinic. And our thyroid program, we're looking at doing the same thing. We're working on first-in-human regulatory documents. Our clinical and regulatory team is actively working on that from at this point in time. So we have a lot going on in the company, and we are also involved in a lot of business activities and discussions that are moving forward. So I would like to stop there, and then we have a list of questions that have come in that that I'm going to give you a little more flavor on in terms of to get some answers to investors.
Thank you very much, Dr. Tolikis, for this great presentation. I'd like now to move to the Q&A period. I would like to thank all of you who submitted questions, and we will try to get to as many questions as possible. First question, we noticed a lot of activity at Sernova continuing in spite of COVID-19. What provisions have you put in place, and how is everyone coping? How are you continuing to move ahead when many companies are shut down?
So first of all, I would just like to say that Sernova is looked at upon as an essential service, as a laboratory that is developing medical products. We have been allowed by the Ontario government to continue operations. That being said, we have very strict provisions in place to be able to deal with COVID-19. Our laboratories are at a level where we already have these provisions in place to allow our scientists to work there, and our management team and other individuals that are able to work at home have been working at home. And the interesting thing that we're all finding is that with the idea that your home is your office, we're getting people working longer hours than I've ever seen in our lives. So we're actually having to help convince our employees to take some time for breaks and enjoy the sun a little bit. The other part of this is that we, from the business side of things, we've seen the world change and adjust very, very rapidly. So we're doing a lot of Zoom calls with our pharma partners, with our investors, and a lot of teleconference calls. And internally, we're also working with our staff on a daily basis to make sure that we're very, very tightly connected. So the reason you're seeing all these activities is because Sernova is moving ahead more rapidly than ever. And I'm happy to say that we have no COVID in our company or our families, and we're going to continue it that way.
Can you please provide some information on the importance of today's press release announcing the collaboration between Sernova and Ajax?
Okay, so first of all, these collaborations and agreements that occur, you see them come out in the press release, and people probably go, oh yeah, whatever, another collaboration or whatever. But these things take months and months of discussions and working together with the other teams behind the scenes. And then there are significant documents that have to be put in place to determine how we're going to be working together and that sort of thing. So what I'm really excited about Ajax is that they have state-of-the-art technology. And you might ask about what is this about? And it's for to be able to work with us, and we're to be able to take this technology and put it into our starting cells. And it's a genetic modification of the cells. And it allows the cells, if everything goes well, to be able to be blind to the immune system. So the idea is that if you look at our diabetes and hemophilia cells, we are looking at the potential of being able to take this technology, put it into our cells, and then have those cells go right into our cell pouch to be able to connect up to the blood supply and then being able to produce their product into the bloodstream, directly put into the cell pouch. So this is a very, very advanced technology, and the founders of Ajax were the founders of Geron. So if you look up that company, you'll see that they have very, very wide experience, and our scientific staff, who was very, very experienced in the regenerative medicine field, is really excited to be working with the Ajax team. So look forward to seeing more updates on that program as things are going forward. And the other part of this is that Sernova is looking at different kinds of local immune protection technologies. And so we are being very, very aggressive moving forward in this field. And the exciting thing here, again, is that it's going to allow us to move away from the use of immunoprotection medications to a world where patients will be getting the cell therapies without the need for any medications. and we'll be able to treat a much, much broader array of patients. Okay.
Let's move to questions pertaining to the University of Chicago clinical trial. Can you update us on the status of the clinical trial, especially given the added COVID-19 issue?
So first of all, I just want to give kudos to our clinical team and who has been working very, very closely with the CTI team. So these COVID issues have come up very, very quickly, right in the middle of our clinical trial. And our team has worked very, very hard to be second to none in terms of ensuring that our patients are safe and that our program is moving forward. So we have been following the FDA guidelines and the World Health Organization on COVID. Our plans are to continue right now. What we're seeing is that the University of Chicago Hospital is starting to lift restrictions, and our clinical investigator is looking forward to moving forward with being able to bring our patients into the hospital for additional procedures as required. And our patients are located around the United States. And so we've been working very closely with these patients to schedule on-site required visits when they are able to occur. But what we've also done is work with our clinical team to be able to make sure that patient assessments are getting done within their communities. And there are ongoing and frequent calls with the patients to make sure that they are safe and that they are getting their safety precautions. So I'm pleased to say that all of our patients are very happy with the processes. They're all safe, and the study is moving forward as we can.
How is clinical study data that needs to be collected being handled?
Yeah, so I've answered a little bit of that question. And what we're doing is we have it set up so that patients can go to local labs as required to get blood samples and collect data as required. And then those patients also are being routinely checked by what we call telehealth visits, right? So they're in communication with the physician as needed, and all of the data is being collected and inputted into our clinical study database through the clinical site that then goes to our CRO in that area. So additionally, as we discussed before, the patients are wearing continuous glucose monitoring systems, and they also are continuing to be used, and they're updated to the data is being updated in a blinded fashion to our clinical study site and database. So things are moving ahead from that perspective. And again, just want to say that we are continuing to collect data.
Will patients have the cell pouch and islets longer than in the timings initially presented?
Yeah, so that's a really good question. And you know that in Canada and the US, a number of hospitals, the first thing they did is shut down their hospitals essentially for surgical procedures and stop visitors from coming in. If you have relatives, you know that this has happened. And so we're really lucky because we already know that cell pouch, like if we have to wait a little bit to take a sentinel device out, that's not a problem for us. We're just getting longer term data on our patients. So we're going to see a little bit of shifting there, but we're not worried about that. Also, with patients that have been implanted with pouches and they're waiting for cells or for islets, we know we have already done long-term studies to show that cell pouches remain highly vascularized for a very long time. And we have the ability to be able to wait for a period of time to be able to put islets into the cell pouch. So this is not of a concern. And again, our physician is very, very committed to moving the study forward and keeping everything going because he feels very positive about the work that's being done right now. So as the hospital opens up and allows more procedures to occur, then you're going to see our patients being able to get in. So this is not a big problem for us.
Are there any special measures for the patients and their safety?
So as we said before, our clinical team's been working very closely with all of the FDA provisions and also with CTI, which is our CRO, on education of patients and making sure that they are safe, that they're getting their proper medications, and that there is a lot of communication with the patients. So we, because we're taking special care of the patients, we don't see at this point that we have had any issues with patients so far from a risk perspective, because we're mitigating those risks ahead of time. And the other part of this is that the monitoring and safety of our patients is utmost important to Sernova, and also the physician who interacts with them very, very closely. So this is a very, very, when you're in a clinical study like this, it's a very tight team that forms together, and that's what Sernova has developed.
You've addressed it a little in the presentation, but can you address Sernova's patent portfolio? Where do we stand with our patent protection?
Yeah, so patents are very, very important to the company. And as I mentioned in the presentation, our patents are very, very broad. We have broad claims, which allows you to have that control. And then we also have patents around our cell technologies, around our device technologies, combination product, and also we have additionally filed additional patents more recently, which gives you another 20 years of coverage. So we're also working what we would consider the best patent lawyers in the United States, which are out of Boston, which is one of the biggest technology centers in the world. And so we've been very, very successful at not only getting patent claims around the world, including Asia, China, North America, South America, Eastern Europe, and Western Europe, but we have now been filing additional layers of applications and getting patents on broader and broader array of our technology. So we're really thrilled about this. And we have actually been evaluated by one of a very large company that we're interacting with, and they have looked at our patent portfolio. And this is important because of our interest in licensing, and they have approved essentially the quality of our patents and validated that.
What is Sernova's partnering strategy?
So again, we are working, we have our own technologies. So we have our own stem cell derived technology for diabetes. We have our own cells that we're working on for hemophilia. We have our own opportunities for thyroid disease. So if we end up not partnering, we are a completely independent company that can develop these products all the way to the clinic and to the market. However, we are also interested, and we see a good opportunity to be able to work with large pharmaceutical companies to be able to advance our programs more quickly, and for potential licensing deals and collaborations from that in that way. So we have been very, very active with working with other pharma companies, and you can see that we're not putting a lot of press releases out about this. What we have found is that a number of times pharmaceutical companies like to work with companies like ours and keep it quiet, so that just for competitive intelligence reasons and that kind of thing. And then all of a sudden what will happen is a deal will get done, and it will look like it happened overnight. And we always talk about the idea is that the overnight deal that took 10 years to get to. So I can tell you that we have very, very strong business relationships in the world. We have super high respect from the pharma companies that we are working with. We have done collaborations. We've gotten outstanding data, and stay tuned going forward. And as our clinical trial moves forward, our technologies advance, our local immune protection technologies, this company is getting stronger and stronger, and is a company to be reckoned with around the world.
I'd like to thank you, Dr. Tolekis, for this update and for taking the time to answer the questions today. And I'd like to thank all the participants who have joined us today. As a reminder, a copy of this presentation will be made available shortly on Sernova's website. Thank you very much to all. Stay safe and healthy.
And thank you very much.